Good afternoon. We will get started. So I have two announcements to make. Those of you who are either listening or here who are taking the course and want to receive a certificate that you've done well, the final exam will be posted probably the middle of next week on our website. It's a multiple choice exam. If you've attended 50% of the sessions and signed in and you pass the exam, you'll get a certificate, which we hope will please you. It's not something that you need to sit up and worry about. You can take it more than once. Most people don't have to take it more than 2 or 3 times, so don't worry about it. The second thing is that Tony Fauci was to have spoken here last Tuesday, but he had a higher calling from downtown, which conflicted at the last minute, so there wasn't much we could do. But he was very anxious to present his ideas about evolving global infections. And so he's going to speak next Tuesday, which will be the final session of this course, and it'll be suitably posted and everything else. And we encourage you all to come. Okay. So, all right. So I see a few new faces who I don't think have been here before. So pardon me if I indulge for a few minutes. Is there anybody who doesn't know what that picture is? Don't hesitate. Raise your hand if you don't know. Ah, Dr. Rapoport, you don't know what that is. Okay. Would you please tell Dr. Rapoport what that is? That's the Brooklyn Bridge. And now, why would we show a picture of the Brooklyn Bridge in a course entitled Demystifying Medicine? Because we are like those two gents on the catwalk, halfway between Brooklyn and Manhattan. Well, make believe on one side, that's Manhattan, the old Fulton Fish Market in the distance there. And make believe that that's clinical medicine. And then make believe on the Brooklyn side are all the incredible advances in basic biological engineering, mathematical sciences, and so forth. And there's a huge gap. So one of the intentions of this course is to try to play some part in bringing together these, what could be very separated areas. And particularly for postdocs and PhD graduate students here who ask me, they're all interested in, careers and how can we do something that influences human health. So part of the purpose of this course is to introduce you to major medical problems. And what are the big unanswered questions? And maybe you can figure out where, with your knowledge, particularly of more advanced reductionist science, there may be something that you could contribute, not the least, to know who some of the clinical people are, and you can communicate. Okay. So today's topic of schizophrenia has been described by many people, particularly our first speaker, Judith Rapoport, in terms of it being a devastating disease. And I put it in the parentheses, or diseases, because I want to make the point. As has been illustrated in virtually every disease session we've had in the course, it invariably starts out in medicine with a clinical description provided fundamentally by clinicians, later aided and abetted by epidemiologists and maybe pathologists. But it's a clinical description. And very often that description is the tip of the iceberg. And then much later, various aspects of more, call it basic science, come into the picture. And we realize that what we saw before as a clinical entity with variations is much more complex. And today's topic is a good example of that. The clinical descriptions by Kreplin and others, who first called it dementia precox, but later the term was changed because not everybody is demented, and it isn't always in a precocious situation. I was fascinated in reading something about the history to come up with the fact that T.S. Eliot, of all people, is given credit for having said that schizophrenia means a split personality. I don't know if that's true or not, but the book seemed to be very authoritative. But when the description of the disease in its most overt forms goes way back in history, and the response to it, was to lock people up, sometimes to shock them, initially the famous snake pit analogy. And those things go on today in large parts of Africa. Diseases like schizophrenia are not treated by psychiatrists or pharmacologists, but they're treated by prayer camp type setups, which use very similar forms of what we might call terror kinds of treatment for people who are unfortunate enough to have schizophrenia or some diseases. In the Western world, things began to modulate a bit. And so in France, you had Parental, in England, Wilberforce, in the United States, Dorothea Dix, who said, wait a minute, we shouldn't be locking all these people up. We should make them comfortable. We should deal with them. We should work with them. This was a tidal wave of a change. But while that was going on, it seemed to me that classifications were changing. And they certainly changed during the cognitive era, as exemplified by Freud. And then much later in the 20th century, when neurochemistry, I'm thinking of people like Saul Corey and Heine Welsh, Stanley Rapoport, who contributed in this field, who's with us today, and others who contributed a great deal toward trying to analytically determine the composition of the brain in different mental diseases. But this was not dynamic, and it required the arrival of imaging and the whole neurobiologic explosion. So where are we? The questions that pop up in my mind, and I share them with you, and I trust you have others, which I encourage you to ask of our speakers. Can we reclassify diseases like schizophrenia and other mental illnesses on the basis of genomics? Can we identify environmental as well as genetic factors that play some part? And what's the relationship of the diseases to the treatments that we give? Where do we stand with regard to all of this? And surely this is a major public health problem. And our first speaker, Judy Rapoport, is chief of the child psychiatry branch at NIMH. She graduated from Harvard Medical School, did clinical and research training at the Mass Mental Health Center in Boston, Children's Hospital here, and at the Karolinska Institute Hospital in Stockholm. She has an extraordinary book, which I read and would encourage to you, about attention deficit hyperactivity disorder. And she's also written about obsessive compulsive disorders. That's the one that I read. Now, over the past years that she's been here at NIH, one of the things that she has done is to investigate the problem of schizophrenia as seen in very young children. The pediatric, if you will, schizophrenia. Is that different from what you see in older people? Are there markers? Are there clues? Is it the same process? And how does brain development participate or take place in the presence of an illness like childhood schizophrenia. So Dr. Rapoport is going to speak first, and then she'll be followed by Josh Gordon, who's the newly appointed director of the National Institute of Mental Health. Josh got his MD and PhD degree at UC San Francisco. He was studying for his PhD plasticity in the mouse brain, particularly regarding the visual system. And then he went to Columbia University for psychiatry residency and research fellowship, and has done remarkable work on the role of the hippocampus. And in fact, what he has utilized, and I trust will speak to us today, are genetic models or transgenic models in mice which carry mutations that are of relevance to psychiatric disease. So we look forward to both of your talks, and I'm sure it'll be a very exciting afternoon. Judy?
Well, thanks very much, Wynn, and a pleasure to be here, and welcome everybody, seen and unseen. I gather people are watching long distance. I'm going to talk to you about our studies which have taken place over the last 20 years, and perhaps the most important point I can make is that this is a very unusual angle on schizophrenia, that National Institute of Mental Health has had major schizophrenia programs since the intramural program started, and that if there's one take-home message that would be the most practically useful one, is that stratifying by age of onset is a very interesting scientific thing to do, but that unlike obsessive compulsive disorder that I thought was rare when I started studying it, and it turned out to be surprisingly common, in contrast, I thought childhood onset schizophrenia would be rare, and it turned out to be much more rare than I ever thought. So that in some ways I'm a very odd person, and I want to make sure that the message gets across that almost everybody diagnosed with childhood schizophrenia has something else. And it's the only time in my many years here in the intramural program that I was worried that studying it could lead to misidentification, as I will show with you. So what I really want to talk about is what the clinical and neurobiological features of childhood onset schizophrenia are, because as you know, through studies of Alzheimer's, studies of diabetes, et cetera, there are many cases across all of medicine and pediatrics where having a particular age of onset clarified, perhaps had a more homogeneous population, and that was my hope. Also, the intramural program is the only place. Can you imagine trying to get a grant and saying it's going to take me 23 years to get a decent enough sample? Not going to happen. And so to the extent that one should focus on things that you couldn't do anywhere else, this certainly fills that bill. What we were, of course, interested in was the ability to study brain development across childhood and adolescence prospectively. And it is in the intramural tradition that you have perhaps a relatively smaller group of people, even after 20-odd years, but that you can study them in a much more intense way and in much greater depth. By the way, if people have any questions or clarifications, I much more prefer to be interrupted than waiting for the end, so feel free to do that. So the description of schizophrenia in the DSM would be that you need to have at least 2 of hallucinations, delusions, disorganized speech, behavior, negative symptoms. And the negative symptoms really mean a kind of lack of volition. You can say to the person, you realize if you don't do your homework, you're not going to finish high school or you're not going to finish college. Don't you know that? And they say yes. But it's hard to see that they just don't care. And obviously you rule out other disorders such as depression and so on that would account for this. And we have searched for children who have this, but much more common, and about 1% of the world, pretty much worldwide, in very different types of populations and countries do have schizophrenia, and it is not a rare disease. And because of the severity and chronicity of it, is an enormously expensive disorder, particularly in developed countries. This may be one of the few disorders where the not developed countries may be more humane and have familial care. And it's much more of a problem, I think, caring for in developed countries in some ways. So this just makes the point over again. And this could have been a study. There have been very similar rates. And this is age at admission in countrywide. There's one for France that's similar and so on, versus the rate per 100,000. And there's several things that you can see. One is that, interestingly, in the early 20s, late teens, which is when the more typical and noticeable age of onset is, there's a much higher rate of males. But if you get down to the vanishingly rare rate, which we studied, defining our population of age of onset by age 13, for some reason that sex difference disappears. It's also very clear from here that this really is rare, at least by reports, and I can attest for the fact that probably many of those had something else, even the ones showing up on this graph. So I'm going to just give you an example of a patient, and this is a patient who has a very good example of flagrant thought disorder. And by age 10, she started having very abnormal thoughts and was exactly the same way pretty much as she is at 14 in this brief videotape that I will show you, which is an example of the flagrantly disordered thinking that can happen in schizophrenia. I've obviously picked an extreme case because there are other subtypes. For example, patients with paranoid schizophrenia who learn very soon to keep their delusion secret. So this is one of these very clear cases, but they're not all that clear. So what do I do to start this? I don't want to go. How do I get this actually started? I don't know how to get the, thanks. Thank you.
Tell me what you'd like to get at the store, Shai.
I don't know.
You could get anything you wanted down there. If you had $1 million. A what?
Guy screwer.
A guy screwer?
Yeah.
What's that?
Because I'll make it this thing about pregnancy.
Mm-hmm. What kind of thing?
He has a light chance of marrying.
Is it a book?
No.
No. It's a little kit or something?
No.
No. But it's something about pregnancy.
Is it a real tuna fish?
What do you think?
A mermaid girl? A medium one? I mean, a small one?
Small mermaid
Not
girl?
a medium one. I've seen a large one. Like on Splash. When I seen that this part was just tuna fish, this small cup tuna fish.
When there was a mermaid, what did you think of that mermaid on Splash?
She's better than any other mermaid I've ever seen.
Is that right?
Mm-hmm.
I saw that movie.
Anyway, I think you get the idea. We didn't try to capture negative symptoms because that's a very boring film indeed. You really just see someone sitting there while they're often not answering questions, and but makes a very boring short film. So part of the problem in studying schizophrenia is that the word, this is biology, there's no ors, there's only ands. So this is my picture of all the risks and every single one of these risks have been proven and validated many times. The problem is in biology that they're all there, they're all documented, and how to put it together in a meaningful way is an enormous challenge. Now, because we went to all this trouble, and as I say, we had to recruit nationally and include Canada in order to end up with 137 bona fide cases 24 years later. I want to hasten to assure you we were doing other things at the same time, but the accrual of about 7 patients a year was not for lack of trying and not for interviewing hundreds and hundreds of children. But what we actually found in one picture was that it's much too overstating it to say it's more homogeneous. The note that Wynn made in his introduction as are these multiple diseases certainly still stands with us, but that we did have some evidence that this is a particularly brain vulnerable population to what makes you get this disease at an age when it really shouldn't happen, that possibly there's more an exaggeration of the normal brain development across adolescence, which I will show you, because we had the luxury of seeing many of these patients every 2 years for up to 10 years. And so we have movies, so to speak, of brain development. And then what we have very strikingly is a very high rate of premorbid disturbances, but a wastebasket of premorbid disturbances. We ruled out anyone who had an IQ below 70. So this is not a study of intellectual disability, although children had a lower IQ than the average age adult group. But we had a much higher percentage than seen in adults of pre-psychotic, premorbid developmental brain-based disorders, and that was also very striking. So some more specifics. But everything you see on this slide is unquestionably replicated many times in adult populations too. So I could spend the entire time just on this study, just on this slide, because we looked, for example, at what would be exaggerated. Was there something going to leap out in many of these things? And the left-hand side were things that we had no strong indication that this was really salient and early onset. And the reason we're looking at those left-hand things is because they've been proven without a doubt to be risks in adults. And we're not saying they're not risks for child, but that's not why we conducted probably the world's most expensive schizophrenia study, because if you're studying in adults, you don't have to go more than a 50-mile radius to get your entire study population. So we had no evidence, for example, of early puberty. That could be one thing you might imagine. Or that the fathers were older paternal age, no thing. Season of birth, particular trauma. I should also say as a disclaimer, when you're studying something very rare, you have all sorts of referral and ascertainment biases that there's nothing to do about, but you have to obligatorily mention this. And the siblings were not particularly dramatically impaired. We also made sure, start turning to the right-hand part of this slide, that they met unmodified DSM diagnoses. And to a child psychiatrist, our mean age of onset, around 10, this is a late onset problem. In the first 3 years of life is when you would diagnose autism, for example. And because we did research, we didn't rule out if someone had a prior diagnosis of autism, which turned out to be an amazingly clever thing of us to have not ruled out, which was good, as I will come to. But all sorts of studies in skin conductance, smooth pursued eye movements, anatomic brain MRI with large ventricles, all of this was absolutely true for the children. We also were very interested because we were one of the few people that could use a drug that has very dangerous side effects, namely clozapine. And I'm not going to show these studies because they were very small, but when we first started our study, many children were on low doses of haloperidol, and we did a double-blind crossover with clozapine. and showed that clozapine was astonishingly, even with very small n, much better. The reason why we fortunately had an inpatient program doing all of these studies so that we could worry about the real issues of EKG, of heart danger, obesity, and bone marrow suppression, all of which are very real and all of which are absolutely worse in children. So this is not a study for the faint-hearted. But to the extent as a clinician that I have a reward out of this study after 23 years as a personal clinician, we ended up with 6 children who could look you in the eye and say they used to have schizophrenia and they don't anymore when they take their clozapine. And amazingly, and this is something like the adult percentage. I'm not trying to tell you that children are more responsive to clozapine, but in adult studies, there's argument that there's a superiority across the room. But having seen these children and know that our own follow-up showed that these children have stayed on it permanently unless there was one who got bone marrow suppression and then we retested him and he responded without it after he was 18. But the miracle to a clinician like myself was seeing these 6 children and knowing from phone contacts that this holds to this day. For the others, it was just a very good antipsychotic, and most surprisingly, many of the families chose to stay on this. For those of you not familiar with antipsychotics, they're the old ones, the original antipsychotics, which were effective particularly for positive symptoms, but were problematic because of tardive dyskinesia. And then there was a second generation of antipsychotics. And then clozapine is in a class by itself because it picks up a handful of patients that nobody else does. And we have studies pursuing this and what way these 6 patients are different, together with Novartis, and looking at fibroblasts and neurons and seeing what does clozapine do to their neurons as opposed to the others and so on. But I am not, yes?
Do you have issues with weight with clozapine?
Yes, one of the many bad things with clozapine, and it certainly shouldn't be a first choice drug, but if you have the opportunity with experienced people, I think we would advocate for the juice, because on the average, most families preferred it whose trial did not get. And it is interesting that there was a substantial subgroup, maybe 25% who didn't gain an ounce on clozapine. So there's all of that to it too. At any rate, and we had, for those who could tolerate clozapine, which is more expensive, which requires a monthly blood test, we have over 70% long-term compliance. And I mean 20 years. We've contacted everybody. And so there's something about it that seemed better because, and several of them had to try other things for a while because, their insurance company wouldn't cover it and so on. And what that mysterious thing is, but it's really for those 6 children that it was the miracle. Okay, so as I say, the phenomenology of the children we see, they very much looked like poor outcome adult patients, and by and large had what's called an insidious onset. They didn't have an acute break. It took place over, at least 6 months. And what was very interesting was that the premorbid disorders weren't any one thing. They just suggested that there's this really widespread abnormalities in brain developmental processes. So you see, this was, we spend 2 full days in outpatient screening, and you can imagine what some person in private practice does who never can see a patient off drug, who has an hour or 2 at best. But at the end of 2 days of screening with structured interviews and cognitive testing and long structured interviews of everybody, we had about half after 2 days we thought they could still perhaps have schizophrenia. But it's clear that psychotic symptoms happen in children with a wide variety of other reasons. For example, in depression, 1/4 of people with bipolar disorder have very clear psychotic features as part of the disease. Often, the people who were sent to us by mistake would have a combination of very poor attention span, together with enormously extreme anxiety. And if you take many other diagnoses and you make them very, very extreme, someone who is at the very extreme end of the anxiety disorder can sound like they have a thought disorder. At any rate, so we had about half who we went on to enroll in our program. And this half, after inpatient admissions, these were patients who also included a up to 3-week drug-free observation. And it is a very sad commentary on American medicine that this is not financially possible for most people, nor is observation drug-free, because the reason why most of these people got eliminated was because we had the luxury of inpatient observation and drug-free admission. And this is really very sad. The drugs can mask bipolar disorder, which got clear for many of them. You can have psychotic depression, and several of ours really had mostly a form of autism, very severe anxiety, as I mentioned. And so we ended up, it's a few more now, it's 137, but were actually admitted really in count in our study. And what was interesting is that what you're supposed to pay attention to is that our mean age of onset is about 9, but they were first evaluated as an average at around age 6 for this wide spectrum of neurodevelopmental problems. And what you should know in terms of the adult studies, this is one of the more so points I'm making, because in cohort studies done with adult schizophrenia, the famous one in England, everyone born in 1944 and followed prospectively, what they find is the ones who developed schizophrenia later, they talked a little later, they walked a little later, but they were within the range where the pediatrician would say, and say appropriately, this is within the range of normals. What's different about our patients was they were seeing therapists for their language delay, for their clumsiness, for their lack of ability to learn to read. But it wasn't one particular thing. And also, child psychiatrists use low dose of antipsychotics very widely for behavioral problems. So many of these children became schizophrenic while on low doses of antipsychotics. It wasn't clear that earlier treatment of drug treatment would have stopped anything, quite the opposite. So this is what I'm trying to say, that we did have about 13% of our subjects for whom it really was a surprise. If you had met their parents before they were 10, they would have had no special worries or concerns. They had friends. They seemed like typically developing kids. But for the others, between educational, language, social, motor development, and to our astonishment, 20% of our cases did have autism spectrum disorder. This gets very interesting when you hear about the genetics. So that's a point I'm making very strongly here, because the non-specificity of genetic risk we've gotten used to it by now, but when I first started studying this, I found that astonishing. And maybe it's not going to be such a bad thing, having a non-specific risk in terms of the potential for future prevention. But anyway, I was amazed at that we had 20%. So a typical one in that green area would be someone who at age 2 or so, parents, they clearly had no social mutual interaction, and they had problem with language, and they came to an autism clinic, and they'd be spinning and flapping, and they were absolutely typical autistic kids. And at least the ones we saw got better. And then around age 9 or 10, they started to have delusions and hallucinations. And they went back to the autism clinic and they say, we don't know what you have, but you're not one of ours. And in most of these cases, the actual autism spectrum was no longer their focus. And they had improved quite a bit on that. And this was clearly a new and totally different disorder. So that's, but the main point of this slide is the non-specificity of the concept of just brain vulnerability. And then, as I said, we had the luxury of doing brain imaging on these cases. And we were very interested in timing. And Jay Geed in our group was doing a very large normal control group that was timed for the every 2 years when our patients could come back. No easy feat because they lived all over the United States. There were only a handful within a 50-mile radius. And so we were able to study over time how gray matter developed. So let me start this. I think if I start this one, yes. So what you'll see is during childhood and adolescence, there is a process. Just look at the one on the right-hand side, and that's turning blue. And everybody in this room, over the course of their development, this is what happens to your brain. And as it prunes down, all the MRI shows is gray, white, or different shades of gray. But the point is that when it turns blue is when it reaches the maximum where it's going to be during development. And then this process really slows down by about 19 or 20. Now I have this color-coded because the map on the left-hand side is the difference between the child schizophrenics and controls. And I have to have it color-coded because it looks like the same pattern. And if I didn't know which one, I have to make it red so that I know which one is the schizophrenia group. And the point is that this was one of our more so's, that they seem to have a stronger process and a shift to the left of this. It was happening earlier and more so, okay? So that was one of our anatomic findings from being able to image and re-image these children. One of the things we were able to do was a resting state connectivity. And I don't want to spend a lot of time on this because these slides get complicated. But in collaboration with Alec Martin's group, we used 26 regions of just the brain at rest. We have kids trying to do tasks, but so few of our cases were able to do tasks. I'm going to skip over the task-driven ones. But what we had was we ended up with clusters of, and let's just call it a red cluster of social brain regions in the red cluster. And the green cluster are the basic things of sight and movement and hearing and so on. That's much more basic kinds of functions. And what we showed is that there's pretty good correlation within the network, but the point of this, and I don't want to take that much time from this talk to go into it, but the biggest thing was that the connections between the social emotional cluster and the very basic hearing and seeing ones are what's missing. It's the connections across these two clumps, which was where that seemed to be. And this correlated with the degrees of severity and so on, this degree of this disconnectivity. And I'd say across imaging, and as I say, I consider myself in many ways an outsider to the schizophrenia field, but I think that the disconnectivity has remained a major concept in many, many studies in adults and possibly has some bearing on future treatments. So this is not a special thing for the children, it's just this came out rather clearly. Then the other area in which was more so that made us glad we had gone to all this trouble was it, quotes, more genetic. And genetics is changing so fast that what we mean by more genetic changes every day in terms of what the possibilities are. But we looked at just obvious things, and atypical psychosis as an indication just for looking at chromosomal screening, the old-fashioned kind. And we found that we had 14%, almost 15%, had a copy number variants, which are a piece of a chromosome that you have either two of, or you have either more than you should have or less than you would have, so you can have a deletion or a duplication. And we had a variety of chromosomal abnormalities. We had a higher rate of sex chromosome abnormalities. When I talk about a wastebasket, when I say more genetic, the answer of whatever type you're asking about, the answer is probably yes, because it was true of everything we measured. And so here we have a whole bunch of different things that we found with different collaborations across the way. And it adds up that there were 18% with one of these things or another. And that turns out to be higher than the rate you would see in adult schizophrenia. And so in this very garbled, very general sense, very much like the neurodevelopmental, in that they were different, but is this a more homogeneous group? Not in the way that we might have thought. So we picked 40 neurodevelopmental related that we focused on: autism, schizophrenia, intellectual disability, and or epilepsy, although that didn't contribute very much to it at all. And then of these, we chose 46 loci, and that's how we picked what we looked for. And what we found, to our surprise, was that we had a very high rate of the highest risk known, other than having an identical twin with schizophrenia, which would give you, I think, about 40-something%. I'm not up on the latest, but it gives you a much more than 1% chance. But second to having an identical twin, the highest risk is 22q11 deletion. And I think there's 6 of them here. And that's the highest rate. They're known to be increased in schizophrenia. The rate in the normal population is like 1 in 4,000 is the rate. And I think it's a higher rate in schizophrenia, but probably like 0.5% or something like that. It's not a lot of adult schizophrenia. So having 5% with this is one of the higher possible percents. And then we had a variety of others. This is a complicated slide, but what you're supposed to look at is at the last column. You can see that many of the things we found have been reported in autism spectrum disorder, intellectual disability, epilepsy, and all 3. And that it had no relationship. For example, the ones that had them reported in autism spectrum disorder, none of them were our 20% who had a previous history of autism spectrum disorder. So there was no overlap, for example. So we're talking about very much broader risk in a neurodevelopmental way than we would have expected. And this is just saying that when we compare this to the healthy siblings, having a carrier of what these are just for copy number variants. Even if we leave out the 22q11 deletion, we still have a significant number compared to the healthy siblings. We only had about 70 of them. So these are very small sample studies. It's also true that these are much higher than in autism. We had 2 colleagues that had samples of autism, and they looked for us at our 40 neurodevelopmental copy number variants. And it turns out that the rates are 2-4% of having one of these at least, and that ours were just much higher. So both with respect to sex chromosomes, a variety of other things, copy number variants, and we also screened some of the alternate diagnosis patients who were very severe, very early onset, and we found that they had a rate of copy number variants that was very high. They had a rate of 12.5%. And so these copy number variants, it was a whole variety. Just look at, these are diagnoses of bipolar, schizoaffective, strange psychosis, very extreme hyperactivity, very extreme anxiety. These are patients who didn't function, couldn't go to regular schools, et cetera, et cetera. But they too have an elevated rate. And so what was clear was that, again, that had been reported for these other things. So we got very interested in these. And just to cover the waterfront, we did look at polygenic scores for our patients versus their siblings, and we ended up with a difference, a significant difference. And while the trouble with doing something so rare and so special is we will never be able to replicate this or wouldn't be willing to spend another 20 years trying, but the fact that with such small numbers we have a difference in the common variant score make me speculate that because most of the adult studies had thousands of patients that get significance with the polygenic score. So for that reason, I will cover the waterfront and say this is a, quote, more genetic form of schizophrenia. So, as I'm saying, we've said that this score predicted this, and I think that I've already said we can't replicate that finding. So I've mentioned we have very striking premorbid stuff, greatly overdiagnosed. We talked about the brain development and why clozapine should be tried because it's so amazing. It's worth trying it with the risk because those miracles I will be proud of for the rest of my life. And if there's a few minutes, do I have a few minutes left?
A couple minutes.
Okay. Well, I don't think I'll show any slides on this, but we had an interesting experience, which was that we do share with parents what we find and that these are research findings, and they all want to know, well, what does that mean that we might be a character? What should we test our children? What about the other children? Should they get married? Should they be screened? And we realized that you really don't know much because what the parents want to know is the penetrance of terribleness. They don't want to know that they have a 2% chance of autism and a 3% chance of intellectual disability. They want to know how terrible is this. So a study that I'm very excited about, and I don't have time to show slides, is that we paid for the Children's Hospital of Philadelphia to scan for 5 copy number variants that were very high. And they have pre-scored electronic medical records. And they have 61,000 at the time that we did this. And we looked to see, and the electronic medical records were scored very carefully and included very good behavioral comments and we found that these predicted a lot. It was much more expensive. You had many more diagnoses. You spent a lot more time seeing doctors, and you were much more likely to have congenital abnormalities, brain abnormalities, and we even discovered a new neurodevelopmental syndrome with 22q duplications, that they have gastrointestinal reflux disorder, which is because of the delay in the vagal nerve. It's a vagal nerve developmental disorder because in general, gastrointestinal reflux disorder doesn't happen in controls. But if you're 22q duplication, of which we had loads, you are likely. So this is the first genetic basis for gastrointestinal disorder. Anyway, so we're very into this, totally tangential, but came from putting ourselves in the position of the parents wanting to know the penetrance of terribleness. And I think we're on our way to having some hints and useful hints for them. But I'll stop here and thank you very much.
Thank you. Thank you. So, please.
Sure.
Yeah. So people who are listening can hear the question.
So, having had the opportunity to follow up your sample for many years, when they grow up into adulthood, can they be differentiated between adults with schizophrenia?
No, they can't. They can't at all, except they're very chronic, poor outcome. These are not people who are going to be self-sufficient, have a significant other. This is a very, and I don't know how much is referral bias to an inpatient program across the country, and I don't know how much is referral and ascertainment, but ours were an extremely chronically ill population.
So why did the 6 get better?
What do you say? Why did the 6 get better? I'm hoping that our collaboration with Novartis that has 6 for whom olanzapine was equivalent, and 6 of the trouble with fibroblasts is that they don't grow up very long, and that's a problematic field, but I'm counting on Ricardo Dolmetsch to come up with something. Yes, I'm sorry.
I had two questions. One is, I was wondering what the de novo rate of,
Right.
CNVs or, particular
Right.
mutations may be in schizophrenia. And I guess that's the first question. The second question would be, is there molecular mechanisms that are known to be able to correct or to edit for copy number variation?
Yeah.
And are
I
those
believe
maybe
the answer is yes,
targets?
but I will defer that question to Josh. I think yes, I think CRISPR would do that.
Yeah, there are ways to do it.
Yeah.
I don't know if they've been tried yet.
Right.
So are you asking if they're natural mechanisms or you could engineer something?
Well, no. What I'm asking is, what is known about mechanisms that edit? For example, there's editing for transcription, for translation, but are there editing mechanisms for translocations or for looking for translocation or deletions of chromosomes?
I don't know. I know how these things form, which is that there are these repeats at the ends of the common ones anyway that cause mistakes. But I don't know if they're editing mechanisms to try to prevent them.
And the de novo rate is how high?
Oh, some were de novo and some were not, and our numbers were so small, but being a carrier didn't correlate well with our diagnoses of the parents.
Okay.
That's the trouble with small population studies.
So, may I ask, years ago, there was a Dr. Dick, I think was his name, who had a school called Summer Hill outside of London, where he took very, very young children who had symptoms like the kind that you described. And after about 10 years of their being in that school, according to his book, about half of them returned to fairly normal living.
Right.
Is that
Well, any child that we saw that was below the age of 7, we were not convinced had the diagnosis. We were referred many cases. They had something else. So if they were very young, we probably would have, really young, we would have probably disagreed with the diagnosis. But I would be very skeptical with respect to our patients. They had very poor outcome, and I don't think many of them, it sure mattered what state you were in. There were some remarkable schools and state services that helped children adjust very well, as opposed to you don't want to be from Alabama and have this disease. But so it isn't that we didn't see environmental help, but no, we didn't see anything wonderful.
Are there any other questions right now? Well, we'll have time after Josh Gordon's talk. So there'll be time for more discussion. Thank you very much.
Sure.
Thank you, everyone, for coming. And I think you'll find these talks quite complimentary. So I'm going to talk to you about schizophrenia from what I call the genetic and genomic perspectives. And I think you'll see from the message of the talk that these are two different perspectives. And hopefully you'll see why. Is this still accurate? Maybe not. So we'll just go right to the disclaimer that the data that I'm going to show you from my own lab, which comprises about half to 2/3 of the talk, was done while I was at Columbia University. And these are the folks who did it. In particular, I'm going to tell you work done by Torfi Sigurdsson, who was my first postdoc, Tim Spellman, a really talented graduate student, and Makoto Tamura, another postdoc, also talented. So when I started out my career in neuroscience as a graduate student, this might be how we would have described the task that we had before us in terms of trying to understand how genes lead to disease phenotypes in general and specifically in brain disorders and in psychiatry, where you have some mutation that leads to a disease phenotype. But of course, even back then, we knew that mutations don't code for phenotypes. Genes don't code for phenotypes. Genes code for proteins, right? And those proteins are expressed in cells and carry out the many processes that a cell has to carry out. And at least in the nervous system, those cells then assemble into neural circuits. Those neural circuits are assembled into larger components, which you might call neural systems, and it's activity within the context of those neural systems that drives behavior. So if a mutation is going to predispose to a disease phenotype in a brain disorder, then that mutation must act through this process. And one could say that in order to understand how this mutation leads to this disease phenotype, one needs to understand the consequences of that mutation across multiple levels of the nervous system. And I should point out that this task was in and of itself daunting, and this I'm going to call the genetics, if you will, of schizophrenia. And the idea is we should find some high impact mutation and study its impact across multiple levels of the nervous system, and hopefully get to an idea then of how the disease phenotype arises. And just as an aside for a moment, I think that may not be immediately obvious why we would need to do this. Why not just characterize the mutation, the molecule, develop inhibitors and modulators, et cetera, and test them clinically? And well, first of all, we knew even at the time that I was a graduate student, which was several years after the cloning of the Huntington mutation, that knowing the mutation did not lead to necessarily druggable targets for brain disorders. And secondly, we don't know in psychiatry which one of these levels is going to be the most efficacious way to treat our illnesses. In fact, in psychiatry in particular, we have treatments that really affect cellular processes like transporter uptake inhibitors or receptor blockers. We have treatments that work at the level of the system, like deep brain stimulation or electroconvulsive therapy. And we have treatments that work at the level actually of behavior. There are many efficacious psychotherapies that work at that level. So we don't know which level of understanding is actually going to lead to better treatments. So it's important to study it across the different levels. So I'm going to tell you really a just so story that comes out of my own lab. I'm going to skip over many of the details, which I'm happy to fill in for you if you'd like through questions, where we try to understand, this process in a reduced preparation, but in a mutation that we think is relevant. And that mutation's already been introduced, thankfully, by Judy. It's the 22q11 microdeletion syndrome. So this is in most cases of schizophrenia anyway, a de novo copy number variant, although I've been told recently that actually you can sometimes find it in the parents. And it's when you're missing one copy of this segment of your chromosome, you have a very high penetrance for schizophrenia. And anywhere from the figure on this slide at 25% to upwards of 40-45% when you follow them prospectively will develop a psychotic disorder. In most cases of 22q11, that psychotic disorder has a sort of normal age of onset, although you already heard from Judy that especially if you follow kids prospectively with this disorder, you find psychotic or quasi-psychotic symptoms earlier. And about 1%, give or take, of schizophrenia cases have this microdeletion. And that's actually a very nice clean multiplication of the penetrance by its incidence in the population and the known incidence of schizophrenia. So 1 in 4,000 live births, as Judy mentioned, have this microdeletion. It's the most common one that we have. And it's also, as Judy mentioned, the strongest single genetic factor. Importantly for the work I did, it's associated with working memory deficits, and that'll become clear why I mentioned that in a moment. So, from the perspective of a neuroscientist, I wanted to get into the nitty-gritty across these multiple levels of the nervous system, and in order to do that, for a genetic syndrome, I wanted to be about the genetically tractable organism. And fortunately, also, at the time I was starting out as a faculty member, colleagues at Columbia had already built a mouse model of this microdeletion syndrome. So these are the genes here on chromosome 22. All but 2 of them lie right next to each other on mouse chromosome 16. And so Joseph Gogos and Maria Koryorgu, and I should mention numerous other labs actually had separately built a model of the microdeletion which lacked this core region of about 20 some odd genes, representing about 1.5 megabases of the mouse chromosome 16. And, so we could have asked the mice if they hallucinated, but it's very difficult to get them to respond, it turns out. So we instead wanted to study a behavior which was practical to study in mice, and for which we had some understanding of the neurobiologic basis, so we'd know at least where in the mouse brain to look. And we chose this behavior, a working memory task. There were two big reasons for this, or three really. One was that working memory is definitively a problem that's present in most, if not all, patients with schizophrenia, working memory deficits, that is. And in fact, working memory and other cognitive deficits are the prime reason for disability in patients with schizophrenia, especially now that for many, if not most of them, the antipsychotics do a pretty good job of treating their psychoses, although we heard that may not be the case in childhood onset. So that's a good reason to study working memory and cognition in general, working memory specifically. Second of all, this particular test, the TMACE test that I'll tell you about in a moment, have been studied in a number of different mouse lines, and importantly, the neuroanatomy underlying the test was well described. We knew that it required two particular brain regions, the hippocampus and the prefrontal cortex, specifically the medial prefrontal cortex in rodents. And so we set about trying to understand how this 22q11 microdeletion might lead to a deficit in acquisition of the task in the mutant that I'll tell you about in a moment. So first, though, the task. So there's three phases to each trial of the T-maze task. It's obviously in a T-shaped maze. That's a giveaway. The mouse starts in the start arm. It runs down the center of that T-maze, and it's instructed by the presence of a wall to go to one of the two arms to retrieve a reward. It goes back to the start box, and after a delay, it runs down the center of the maze again, and this time it's faced with a choice. Which of the two open arms now do I go to? And it has to remember the relatively simple rule: wherever you went during the sample phase, go to the opposite arm during the choice. So this is a very compelling and interesting task, and importantly, the animals take, the mutant animals here in green, take about 2 days longer to learn this test than their wild-type litter mates at 10 trials a day for the aficionados. So it's an acquisition deficit or learning deficit that these mice display. Once you train them in the task, they can do equally well as their wild-type litter mates. So it's not a matter of performance, but a matter of learning the task that these mice have problems with. So then the problem then becomes not the generic problem of gene mutation to generic behavior, or to psychosis or schizophrenia, but rather this 22q11, or more accurately the DF16 model, the deficiency of the mouse chromosome 16. How does it lead to a deficit in this working memory task? And again, we knew that the hippocampus and prefrontal cortex were involved, and in particular, we knew from clever lesion studies that it seemed to be communication between these two structures that was important. So we set about to ask whether these two structures were intact in the mutant, and the short answer of that was that they generally were. And then we asked the question of whether these two brain regions could communicate with each other to a normal extent in the mutant. How do we do that? We implant electrodes deep in the brain of these animals, in the hippocampus and the prefrontal cortex, and we simultaneously record activity for these two brain regions. The form of activity we record for the most part in the hippocampus is called local field potential, which is like the EEG, but deep in the brain. And it's thought, and actually there's pretty good evidence to support the notion that what the EEG does is track over time the activity, I should say the simultaneous activity of hundreds or thousands of synapses within a small chunk of tissue around the tip of the electrode that we have implanted. And if you are monitoring simultaneously the activity of those hundreds or thousands of synapses in the hippocampus of a mouse that's running around through its world, what you find is that activity goes up and down and up and down and up and down. And it does so at rather slow frequencies here and at faster frequencies. And here's the here's the slower frequency highlighted. That's a purple oscillation. And you can see from the time bar that it's about 8 times a second. It's going up and down and up and down and up and down. And that 8 hertz rhythm is the dominant rhythm in the hippocampus, and it's called the theta frequency oscillation. So I might slip into theta speak and call it theta, and that's now you know what it means. So we have our electrode in the hippocampus. It's recording this theta oscillation as the animal runs throughout the maze. And at the same time, we're recording action potentials from single neurons recorded with many electrodes in the prefrontal cortex, from many single neurons, I should say, at the same time. Here's action potentials from one of those single neurons that we recorded from a mouse. And what you can see is that action potential, the action potentials from that neuron tend to fire at particular phases of the theta oscillation. You'll know that these spikes are happening on the ascending phase as that purple goes up. And so we say that this spike appears to be phase locked to the hippocampal theta oscillation. We can do better than appear. We can quantify it. So here's lots and lots of spikes, all recorded from one neuron in the medial prefrontal cortex. And you can see that these spikes are more likely to occur at particular phases of the hippocampal theta oscillation, and in particular around 90 degrees of phase, which is on the upswing of the theta cycle. So we say that this cell is statistically significantly phase locked to the hippocampal theta oscillation. And if we do that for lots of cells in the prefrontal cortex, about 60% of them are phase locked. So this is our measure of the degree to which these two brain regions are communicating with each other. And you have to take my word for it, but although I'm happy to explain why, but I want to skip these in the interest of time, that it's a directional interaction. That is, that the hippocampus sends information to the prefrontal cortex. And that this measure of synchrony, this phase locking measure, is measuring the strength of that communication. Now, importantly for us, the strength of that communication is modulated by working memory. So here's data from the sample phase in orange and the choice phase in the working memory task of wild-type mice. And what you can see is that there's stronger phase locking in the choice than the sample. This is the subtraction on an individual animal-by-animal basis. So in the working memory task, as I told you before, we know anatomically these structures have to work together. And here's evidence that physiologically they actually do work together during this task in wild-type animals. And this, I should mention, is work that Torfi Sigurdsson, that postdoc, did, replicating the work of several other labs, most notably Matt Wilson's lab at MIT, who had showed this phenomenon as slightly different task in rats. So we have this measure of the degree to which these two brain regions are communicating. It's enhanced in the working memory task. And then we ask, what about this measure? What's happening to this measure of synchrony, of working together in the mutant mice? And what you see is that the strength of phase locking on a neuron-by-neuron basis is reduced in the mutant animals, and that's true whether you look at the sample phase or the choice phase or through the entire task. So the hippocampus and prefrontal cortex, again, you'll have to take my word for it, they're basically intact in the animal, but it's the connections between them that seem to be at least functionally disrupted. You can think of this akin to an fMRI functional connectivity measure, although the specifics are really quite different. So we have then a systems level phenotype, the hippocampus and prefrontal cortex that can't communicate well together, and we think that's one of the reasons why the animals can't do this behavioral task. So the next question that we ask, well, what's the circuit element that is important for maintaining hippocampal prefrontal connectivity that's disrupted in the mutant? And there we were at a little bit of a bind because the hippocampus and prefrontal cortex, you can send information from one to the other through several different pathways. So we chose the most obvious one and we got lucky. The most obvious one is a direct projection that goes from the ventral part of the hippocampus to the medial prefrontal cortex. It's a direct monosynaptic unidirectional projection. So there are axons of neurons here in the hippocampus that infiltrate directly into the prefrontal cortex, synapsing onto both excitatory and inhibitory neurons in that prefrontal cortex. And so Tim Spellman tested whether disrupting these axons, or their ability to conduct information, would actually disrupt this behavioral task. And I'll just show you one short experiment that he did to prove this, and it involves an optogenetic manipulation. So in this case, what we do is we take a virus that's carrying a gene that encodes for a bacterial protein, ARCH 3.0, and this bacterial protein is very special. How special? Well, when you shine light onto this bacterial protein, it starts pumping hydrogen ions out of the neuron. And so hydrogen ions, of course, are positively charged. So when you pump a positively charged ion out of a neuron, that neuron's membrane potential will hyperpolarize, meaning it makes it more difficult for that neuron to fire an action potential. All right? So we can imagine we express this protein in the hippocampus of the mouse, and if we were to shine a light on those hippocampal neurons, we would make it more difficult for them to fire action potentials and more difficult for them to send signals out, including signals to the prefrontal cortex. But that's only part of the trick. So we can turn off those neurons on and off at will, but what we really want to do is not turn off those neurons, because those neurons in the hippocampus, they send information to all kinds of places. And we don't want to know what the function of the hippocampus is. That's actually been well described. We want to know what the function of the axons that send information from the hippocampus, specifically the prefrontal cortex, is. And that's why this diagram has these little things up here. These are fiber optics, where we can direct light specifically at the prefrontal cortex. So we implant these fiber optics in a mouse that's expressing ARCH 3.0 in hippocampal neurons. Those neurons also express the protein out here at their axon tips. We shine light on the axon tips, and through much work that Tim and others spent a lot of time working on, they were able to show that actually shining light on axons expressing the inhibitory opsin, that actually prevents synaptic transmission or dramatically reduces synaptic transmission. at these hippocampal inputs to prefrontal cortical neurons. And I'll spare you those details. So we now have a method that allows us to say, well, are these axons important for the working memory task? So Tim put the animals into the working memory task, and here's trials where the animals have no light in their prefrontal cortex, and both control animals that don't have our opsin, don't have that bacterial protein, and animals that express that inhibitory protein are performing at about 80% correct. You turn on the light either for the whole trial or the sample phase of the trial, and performance drops. And actually, shining a light during the choice phase had no effect, which was a surprise to us, but it's a little bit of an aside for the current conversation. So now we know that the hippocampus and prefrontal cortex can't talk to each other during the task. We know that this element here, this hippocampal projection to the prefrontal cortex in wild-type animals anyway, is important for this talking together and for this behavior to happen. But what we don't know is whether this connection is disrupted in the mutant. So now we're going to ask the question, what happens to the axons in this connection in our mutant animals? And here again, we collaborated with Joseph Gogos, who had been looking at mutations, particularly in a specific gene, ZDH8C8, which it turns out is important for axonal development. And if you're missing one copy of ZDH8C8, what happens to your axons, and in particular to the axons that go from the hippocampus to the prefrontal cortex, is that they don't branch as much as their wild-type lettermates. So mice lacking now just one copy of one gene within this brain region, they have a deficit in the structure of these very axons that we think are important for working memory. And so we ask whether these mice that are missing just one copy of one gene also have deficits in communication between the hippocampus and prefrontal cortex and in the working memory task, and the answer was yes. So these mice, if you will, are a model of a model. They're a model of the 22q microdeletion, and they have the very interesting coincidence of decreased branching of these, what we think are crucial axons, decreased synchrony between the hippocampus and prefrontal cortex, and decreased working memory. So that leads to an interesting hypothesis. I'm going to come back to that slide in a moment, which is that at least part of the reason that this microdeletion might cause deficits in working memory is that there are deficits in axonal branching in the hippocampal to prefrontal axons that disrupts this hippocampal to prefrontal circuit that prevents these structures from talking to each other, that impairs behavior. So this is a hypothesis at this point. And the question is, how do you test this hypothesis? Now we'll go back a slide. And our collaborators in the Gogos lab, particularly Jun Mukai, a postdoc that worked with him, already had the answer of how we could test this hypothesis, because he showed that you could rescue axonal branching deficits in the single gene mutation by treating with an antagonist to a particular kinase whose expression is disrupted in these developing axons because they're missing this gene. And I won't go through the biochemical details, although I'm happy to tell you about them if you care. But suffice it to say, here's our branching in the wild type, deficits in our single gene mutation, rescue with this drug during development. So the question is, would this work if we treat animals during development? Can we rescue this whole system by, it turns out, inhibiting this kinase with this drug? And so that's what Makoto Tamura, my postdoc, tried to do. He took this drug that seems to work at restoring axonal growth, treated animals every other day during the first 4 weeks or so postnatally, and then waited 3 months without drug for them to grow up, implanted them with electrodes, put them in the T maze, and asked what happens to working memory behavior. Well, here's wild-type animals. It takes them about 4 days or so to learn the task. Our mutant animals, now this is a nice replication, take a couple of days too long to learn the task. If you treat those mutant animals, though, with drug during development, then suddenly they are learning at wild-type levels 3 months, I shouldn't say suddenly, 3 months later. What about synchrony? Here's our measure of the degree to which the hippocampus and prefrontal cortex are talking to each other, this phase locking value. Here's wild-type animals, here's mutants, and here's mutants treated with drugs. So we restore the ability of the hippocampus and prefrontal cortex to talk with each other, and we also restore behavior by simply treating with this drug that restores axonal outgrowth. So we have then a linear story by which we can explain some, and I'll spare you the details, but not all, but some of the phenotypes of the 22q11 microdeletion, particularly the working memory phenotype, by preventing an axonal branching deficit. So we have a hypothetical chain of events with some evidence to support it that leads from the mutation to the behavior. Okay, so this was fantastic. I mean, really wonderful. And it also points to potential treatments, although I don't know how wise it would be to throw this kinase inhibitor at developing humans. And there's a lot more work we would need to do before it became a treatment. But at least it points to, as I said, a chain of events across multiple levels of the nervous system that can explain how a gene produces a psychiatrically relevant behavioral deficit. And so that builds on that promise, that linear promise of genetics that I told you about, which is how we used to think about the genetics of psychiatric disorders before we knew what the structure of those genetics were, which is that we'd have some big impact mutation that would lead to disease phenotype across multiple levels of the nervous system. And so this, as I say, is the promise of genetics. But it turns out that that doesn't really reflect the reality of the genomics of schizophrenia, because this is the reality, and I maybe should have put reality of genomics instead. So we know that 22q11 is an important genetic factor that accounts for a significant chunk of schizophrenia, if 1% is significant. We don't know whether the biology of 22q11 is going to be relevant to the biology of the rest of schizophrenia. But we sure as heck know that the rest of schizophrenia is not explained by a large number of big effect genes like 22q. Some fraction of the variants to get schizophrenia is caused by these large effect genes, but some other fraction, probably a bigger fraction, although there's a lot of debate in the field, is reflected in this particular picture. So this picture, those of you who follow the genetics of psychiatric disorders will understand, is what's called a Manhattan plot. And all 22 of your chromosomes are, plus, of course, the X chromosome, are shown on the x-axis of this plot. Each and every locus within the genome for which you can test for genetic variants is a position along that x-axis. And the bars that you see going up, some are green, some are red, some are blue, are columns of closely spaced genetic variants. And the higher that those columns go up, the higher you see a dot on top of those columns, is the greater the likelihood that that particular genetic variant represented by one of these dots, like let's say that one, is related to the likelihood to get schizophrenia. And this plot is already 3 years old, but it's from a study of some, I think, 30,000 patients with schizophrenia and some larger numbers, something around 60,000 controls. And the newer pictures with double those numbers suggest there's something like 180 loci sprinkled throughout the genome that increase your risk for schizophrenia. The problem is that each one of these loci confers a very, very small increase in risk. So this one here, which is the most genetically powerful one, is your MHC locus, and we think it's because of a deficit in a gene called C4, complement 4. But in any case, this locus confers a 20% increase in risk for schizophrenia. That's just about the biggest one, as opposed to a 30-fold, right? 30-fold of what? 3,000%. So this is a small increased risk. Your relative odds ratio is 1.2 if you have this variant, which means that the vast majority of people who have this variant do not get schizophrenia. And the vast majority of people with schizophrenia, well, I shouldn't say the vast majority. The majority of people with schizophrenia don't have that variant. But it does confer an increase in risk. And we could understand everything about the biology of this particular variant and learn very little about why anybody with that variant has schizophrenia, or maybe a lot. We don't know. But there are all these other variants too, right? So what you end up with, instead of that linear picture I showed, is a much more complex picture where you have lots of different genes over here on the left. And you might as well throw in some environmental factors too, although I don't think we really understand any of them. But there are presumably environmental factors that also contribute to your likelihood to get schizophrenia. And of course, each of those has effects at the level of the cell, at the level of the circuit, at the level of the system, at the level of behavior. And left out of here is the fact that behavior can influence systems function, which can influence circuit function, which influences cell function. Stress can affect the cells, so behavioral. There's basically arrows flying in every single which direction you can imagine. And so this is the complexity that the genomic picture presents. It's not a linear picture. It's hard to even know where to begin to study this from a neurobiologic perspective. One thing we know about this complexity, though, is that there is convergence. That is, for example, multiple genetic mechanisms can converge on single cellular mechanisms. We know, for example, that a lot of the genes that are implicated in schizophrenia seem to be expressed predominantly in synapses. So we can think of synaptic function as an important component of schizophrenia risk. But we also know there is divergence. That is, that a single mechanism at the level of the cell level circuit can create multiple problems, and each with different levels of likelihood at higher levels. So you could have multiple systems problems that emerge from a single circuit function, and maybe it's going to happen in one system and not in another, in one individual, and vice versa. And of course, these divergences also can diverge at the level of phenotype, where you can imagine that the same, say, circuit level phenotype will lead to two different behavior level phenotypes. And we know that as well because we know that the genes that predispose to schizophrenia also predispose, or at least some of them also predispose to bipolar disorder. So we have convergence and we have divergence. What we hope for is that there is some form of critical convergence. That is, that many, if not all, mechanisms lead somewhere in the mid between that gene and the behavioral phenotype. Doesn't have to be at the circuit level. I've just illustrated that here, to some critical convergent phenotype that we might be able to pick up if we could just study the system at the right level. Now, maybe that's hippocampal prefrontal synchrony, or maybe it's the axon outgrowth. Maybe all schizophrenia genes eventually lead to problems with the branching of axons. Or maybe there's 3 or 4 different critical convergences that we need to identify to fully understand all of schizophrenia. Or maybe there is no such critical convergence, and there really are, you know, as was suggested in the beginning, lots and lots and lots of different kinds of schizophrenia. But one possibility and one thing that we want to search for is this critical convergence. And so what that's led us to is really this understanding of complexity that makes you think, you know, we don't need any more a genetic explanation, or I shouldn't say we don't need, because it may be that we can learn enough from the study of 22q and other high penetrance mutations that we can understand enough of schizophrenia to really revolutionize treatment. But I think it's quite likely that we need to take a genomic approach, that we need to understand neurobiology at the level of, you know, convergence from multiple causes and divergence to multiple effects. And that's going to take a lot of different attention. That's going to take, you know, computational people to help us understand what the structures might be. It's going to take analysis of multiple genes at multiple levels and looking for things like convergence and divergence and comparing across models, both genetic models and behavioral models and everything in between. So that's my story of genetics and genomics, and I'd like to say that we licked this complexity, but I think that's one of the reasons why I wanted to take on the job of NIMH director, because I realized I couldn't do it all by myself, that's for sure. Thank you. Yeah. We got a mic, apparently.
In terms of your linear model at the beginning, axonal branching seems to be a very pleiotropic phenomenon to have such a specific manifestation as delayed learning. I mean, wouldn't you expect the effects on the neural system to be much greater, involving virtually all functions?
So one big question is how many different axonal projections have deficits in branching that we've seen. And on the one hand, you're right, because Joseph has looked at least at one other connection, the cortical axons in these mutants, and seen very similar patterns of branching deficits. So from that perspective, yes, you would expect that the system would be affected in multiple ways. And in fact, there are multiple different phenotypes. I only studied one of them, and we haven't looked at how many of them might be rescued by this intervention, or even, but you know, the other thing is that intervention we did, the GSK3, might affect lots of things besides axonal branching too. So we do think it's a widespread phenotype, frankly, and if I look at connectivity across multiple brain regions in these mice, we see deficits everywhere we look. So we think it is widespread. It's subtle though, right? It's not, these axons are getting to the right place. They are branching and they are making synapses. And in fact, if you measure synapses within the brains of these animals, you don't see widespread decreases in the number of synapses. You see very, very subtle, if any, differences in the level of inhibitory interneurons, which have been implicated in schizophrenia. So in these mice, the differences are subtle, and maybe it takes it only affects certain behaviors because those are the ones that are most sensitive. What I can tell you, for example, that I didn't show you, but we've published is that, you know, the hippocampal to prefrontal inputs in this behavior are crucial for encoding new facial information into short-term working memory in the prefrontal cortex. And the mutant mice don't do that as well. They don't have as robust a representation of the goal structure of the environment. So we think, so that might mean that this task is particularly sensitive to this loss of input, but other tasks that rely on that input maybe not as sensitive.
I was wondering, because of the complexity that you just described, and there's even worse is that many of those polymorphisms you showed are actually located in non-coding regions of
Yes.
DNA, and they can work also together in different combinations. Wonder why, using your last part of your argument, if we can understand just a few of the major pathways, why not put more emphasis on families where there is a higher risk and the genes affected there?
So it all depends on your definition of more emphasis. So we are absolutely interested in supporting studies that focus on the more tractable high penetrance mutations of all sorts. And in fact, we have a meeting coming this fall that Thomas Lehners organized to get people together around these rare genetic forms and figure out, you know, how many should we be studying, how in depth should we be trying to study them collaboratively so that we do the similar things in all of them, et cetera, to understand how much convergence or divergence there is from these different high penetrance mutations. So that is absolutely an avenue forward. There is also an argument, though, that this complexity needs to be understood as well, because there may be other mechanisms that are crucial. And, you know, from that perspective, the C4 allele is an interesting one, right? Because that identifies a biological mechanism that might be really important in schizophrenia that is amenable to treatment, perhaps. And there are approaches. The cool thing is that people are developing the approaches to do sort of genome scale neurobiology. They're crude right now, but they're relatively high throughput. And so you can imagine looking at, certainly maybe not now, but in 5 years, looking at, you know, cellular effects of all that variance pulled together by doing high throughput automated, you know, neurophysiology on robot patch clamp cells. Like this is not sci-fi. These are techniques that are being developed. They're being developed to address this problem, which is not only in schizophrenia, of course, but in other illnesses as well. On the autism side, there's a similar, you know, mix of common and rare variants, although they feel like things are more rare, but then other people say, well, the rare variants are more common in individuals with intellectual disability, and you're missing a different kind, so you're missing a kind of autism. So there's an argument, a compelling argument to take both approaches if there's really evidence that the kinds separate into two pools. We don't have that so well in schizophrenia just yet. Although the evidence, they were telling me a year ago that there was more common variant, more of this nasty complexity in schizophrenia relative to the rare. But I think that story is also shifting because we're getting better and better at finding these high impact mutations. So I think the jury's out on the balance of what our endeavors should be.
I had a question. I'm curious. I think my imperfect reading of post-mortem studies of people with schizophrenia's brains suggests that it's dendritic abnormalities that are more commonly found. And then, of course, there was that fibroblast, you know, the neuronal report.
Well, I would argue,
is that found in your mice?
So there are some very, very subtle dendritic spine changes that are so subtle as to suffer from the huge problem you have of multiple comparisons, because they're like a particular kind of dendritic spine in a particular zone around the cell body of a particular neuronal type. And so we need more studies to really understand whether there are dendritic abnormalities. But you have decreased branching of at least of these long-range inputs. We don't know, maybe also decreased branching of local axons, but we don't have the ability yet to really, we haven't yet been able to look at that. So anytime there's decreased branching, you would think anyway there's going to be fewer axonal synapses. If there are fewer axonal synapses, then there damn well should be fewer dendritic synapses, because otherwise things don't add up. So, you know, I think I would say that our data are consistent with the notion that there are synaptic deficits. But you know, maybe they're happening in parallel in all these things, and just so subtle it depends upon which technique is robust enough to pick them up, or it may be that the balance is, that it's an axonal problem in our mice, and the reason why you don't see the dendritic problem is other axons fill in, whereas in humans it's really a different problem, or in the generic schizophrenia, which makes up the bulk of the patients that have been studied post-mortem. Yeah. Thanks.
Well, I'm a little bit conflicted now between both talks, so I have a question for both of you.
Ask away.
One of the interesting take-home messages that I from Dr. Rapoport's presentation earlier was the importance of careful clinical phenotyping and how critical that was over the course of your study. But when I look at this slide and say, well, we're not going to get there just because of all of this divergence at the end, the most careful clinical phenotyping probably won't get us there in terms of identifying the underlying genetics or neurobiology. So I don't know. That's just a comment. If you have any thoughts on that.
$1,000 question. You want to respond?
Maybe Judy first, and then I've thought too. Yeah.
Very briefly, and then I'll turn it over to Josh. One of the things we learned to our astonishment was the high rate of the copy number variants in the people we ruled out, and the fact that the copy number variants that were so high also were markers for something else. And what I'm going to predict is going to happen from these copy number variants is that we will come up with a wide slew of neuropsychiatric patients based on trying to predict from genotype. So with that,
So, so I have a number of thoughts with regard to this. So on the one hand, one could argue that just like we are deep phenotyping the 22q11 mouse, we should be deep phenotyping, and we are, 22q11 humans. And as I mentioned, there are people who are following cohorts of these kids as they age, and they're finding like when they convert to psychosis, and they're actually finding slightly higher rates than published rates of conversion because presumably because they're being followed. It's sort of an ascertainment bias, if you will. And there are consortiums for 22q and some of the other more common copy number variants. It's really hard to do that, though, with many of them because, most of them are not nearly as common as 22q. There's one for autism 16p that's also fairly common. I think it's something like 1 in 10,000 births or something like that. So a little less common 22, but enough that you can get a cohort of a couple hundred if you do a multi-center thing. So deep phenotyping plays an important role, I think, especially in understanding the high impact things. In terms of this complexity thing, I think there's room for deep phenotyping, but you have to think about it on an entirely different scale. So it's going to take a different framework. We could deep phenotype people, figure out their genetics, but we know that we're going to need not tens of thousands, because now with phenotyping you have even more variables, hundreds of thousands of people, right? And we can do that, actually. We can push out, a panoply of web-based behavioral tasks to very large cohorts and ask what the genetic variance tells us about different aspects of behavior. And we can do it in a diagnostically agnostic way, maybe enrich for people with psychiatric disorders, but not care about which ones they have. And also we care about normal variants, because it's quite likely that, and there's some evidence even for the large effect size mutation, these genes are affecting your function on a scale, and if your function dips below a certain level, then you might be labeled as having schizophrenia or autism. That's at least, I should say, one idea. It's one that we're actually interested in pursuing, but it's daunting. But I still think, yeah, humans are the best phenotype, have the best phenotypes we have. If we can find a way to study them, even this complex, we should try it.
Has anybody looked at prison populations to see the frequencies of these variants, and if so, are they higher than?
I don't know the answer to that. I mean, prison populations have very high levels of serious mental illness, so one presumed that but controlling for that, whether there are increases of these high-risk variants, say in what is it called again? Help me here. Personality disorder leads you to get in jail. Anyway, antisocial antisocial behavior, antisocial personality disorder. I don't know.
So let me ask you a question here. This is looking backwards, not looking forwards. in the various clinical pictures that neurologists from time immemorial have described, most of which have syndrome, somebody's name on them, but have been linked in one way or another to the hippocampus and the prefrontal cortex. What can we learn from them that give clues to what genetic or structural changes are present? Do we know anything about that from those standpoints? And I might as well throw in the ultimate one. In the days when prefrontal lobotomies won Nobel Prizes for the treatment of schizophrenia, do we know anything about even the cognitive changes that took place? Do they make sense in terms of what we think we know now about hippocampal and prefrontal connections?
In vague generalities, I would say. I mean, I don't know if Judy would have anything to comment. I mean, certainly sort of temporal lobe syndromes as a result of, surgeries for epilepsy, et cetera. I don't know that anybody said, oh, these folks get psychotic or have working memory problems, actually. But most human working memory is in the verbal domain, and this is one of the details I skip over, but this is not a verbal working memory. It's a spatial working memory, which may be particular to the hippocampus. But on the other hand, from nearly time immemorial, the hippocampus has been implicated in schizophrenia, and in fact, the most replicable findings in postmortem and imaging studies are about the structure and cellular constituents of the hippocampus. And then the second most would be about prefrontal. The latter, well, both of them actually might be looking under the lamppost because so many studies have been conducted in those two structures, at least in postmortem. But certainly for the imaging studies, the hippocampal volume changes in schizophrenia and were, the first thing discovered. I think they were discovered in CT scans, actually, and they've been replicated over and over and over again. Now, what that has to do with the phenomena that I'm talking about, that's anybody's guess. But just vaguely that these structures have been implicated in schizophrenia, whether because we thought they ought to be or from evidence that's been around for a while. I don't know of any specific syndromic data that would point in that direction.
Okay,
Yeah.
whoops.
There's another hippocampologist.
This is a question for both of you, I guess. Yeah. The reason I was asking about sporadic mutations or basically copy number variants is the question, is it possible that the variability that you see is that you also have some kind of like chimeric? So it depends when you get the sporadic mutation.
Yeah.
And has anybody looked
this
at
new somatic mutation business? Not so new, actually, been around a while, but it's come to the fore. People are trying to look at that now in disease tissue, and I'd say the jury's out. I actually was just talking to Chris Walsh about this the other day, and I said, "Hey, Chris, I know you discovered it, or rediscovered it, and it's really interesting and stuff, but is it real?" Which everyone keeps asking me. And basically he says, look, it's real. That is somatic mosaicism in the brain. Yes, we have plenty of evidence that it exists. Whether it plays a role in disease or not is what we need to know, and we don't know that. But yeah, one can imagine the somatic mosaicism contributes to or exacerbates this. Yeah. Particularly if it happened in certain lineages that affected certain tissues.
Judy, you want to add something? Yeah. I have a question to both of you, and part of it is one of the earliest slides that you showed with a high incidence of male diagnosis in young age. And if you think it is real, it is really persistent, and if there is some correlation with sibling and genetic linkage in that population. And the second part is that your X chromosome is very silent. On your slide, there isn't much on that identified, but then we know that the X chromosome contains a number of epigenetic modifiers and chromatin remodelers. So, Judy, maybe you want to address that first.
Well, I don't think anyone really knows. It's certainly been replicated. Everybody agrees about the sex difference, although by the time you reach 45, you have half female and half male. So it isn't like it's more a male disease. That's a strictly age of onset phenomenon. We had expected that to see more males, and we have slightly more males, probably because people are more comfortable about shipping male children to an inpatient ward across the street, but was certainly not anything that you'd predict if it was some sort of linear effect. Not at all. They're pretty much half and half. And I don't think anyone has done very much with this. There's only one group at Harvard that's paid a lot of attention to sex differences. I don't think they've really gone. I don't think they've done much for understanding the disease.
I don't think so either. There's a fascinating hypothesis that's sort of generalized over multiple psychiatric disorders, and it has to do with the neuroimmune interaction. So apparently there's substantial differences in the development of, I think it's microglia, but certain CNS immune system components that are different between males and females that develop earlier in males. And there's this hypothesis that through interacting with those components, you get psychiatric disease certain psychiatric diseases earlier in males. Or that, I should take it back, that the psychiatric diseases that come early are more prevalent in males, like autism. And the ones that come later, like depression, are more prevalent in females. And maybe that's because it's all working through these immune components that are developing differentially. You had something to say about that, Judy? Yeah. I would say it's a hypothesis.
Yeah, there's a broad concept of the developing male brain being more vulnerable. Right. For example, old studies, when you had high bilirubin level, for the same bilirubin level, males were much more likely to develop athetosis and problems. And another study of meningitis, for the same count of bacteria, males much more likely to end up with brain damage. So across all of pediatrics, there's a male brain vulnerability, and that's certainly true in terms of all of the neurodevelopmental disorders, from dyslexia to ADHD, generally speaking, are more common in males. So that's a very general thing.
And that would go along with the idea that when schizophrenia hits in males, it's hitting earlier. I don't know. Yeah. And then the second part was the X chromosome. So I know that there is a confound there with power, because you're much less powered to find associations with the X chromosome than you are with the, what are the other ones called? Yeah, somatic, autosomal, whatever they are, with the other chromosomes. And similarly with the Y, there would be linkage there presumably, but you're really underpowered to be able to detect those things.
Okay, well, listen, I want to thank you both on behalf of everybody and all the folks watching for a very exciting, stimulating presentation.
Yeah, thank you. Thank you for organizing.